Chapter 19, where we will go over the process of embryogenesis. Embryonic development in animals demonstrates many developmental and physiological similarities. This can be seen in the similarities between the human and chick embryo pictured below. Many times these similarities reflect shared developmental pathways. However, these shared developmental pathways can also diversify and give rise to different distinct and specialized body forms. There are many processes that occur during embryonic development. These processes are important to transform the egg into an embryo and eventually into an adult. Many of these processes include fertilization, cleavage, gastrulation, as well as organogenesis. Fertilization involves the fusion of the haploid egg and sperm. This will lead to the formation of a diploid zygote. Two main reactions occur during fertilization. One includes the acrosomal reaction. In this phase, the hydrolytic enzymes are released from the acrosome, which reside in the head of the sperm. The acrosomal process will form to aid in penetrating the egg coat. The acrosomal process has proteins to ensure a lock and key recognition between species. Plasma membranes will fuse and the nucleus will then enter into the cytoplasm. When this occurs, ion channels will open in the membrane and sodium ions will rush into the egg, causing a depolarization event. In many aquatic animals, this depolarization event occurs very quickly, within 1 to 3 seconds post-entry of the sperm. This is considered to be a fast block to polyspermy, or to multiple fertilizations. This phenomenon, however, does not occur in mammals. This diagram represents the fertilization process in the sea urchin. Note that when contact occurs between the sperm and the egg, the acrosome will release hydrolytic enzymes. This then initiates the acrosomal reaction. These enzymes will then penetrate through the jelly coat of the egg. Acting filaments will form the acrosomal process, which will then protrude through the jelly coat. Proteins on the surface will help ensure specificity binding between the egg and the sperm. Contact will then occur and the fusion of the sperm and egg membranes will occur. This fusion will trigger the depolarization of the membrane, which once again acts as a fast block to polyspermy. Note that this is not present in mammals. The cortical reaction will then occur, which we will see in the next slide. The cortical reaction involves the release of cortical granules. These granules will fuse to the plasma membrane of the egg and release their contents between the plasma membrane of the egg and the extracellular matrix. This is usually triggered at the point of entry. This is because there is an extensive release of calcium from the endoplasmic reticulum into the cytosol. This will overall lift and harden the vitelline layer outside of the egg. This will lead to the formation of a fertilization envelope. In a sea urchin egg, this fertilization envelope forms approximately 35 seconds post-entry. This cortical reaction is present in mammalian fertilization. After fertilization, the egg will become activated. Sperm entry will increase overall protein synthesis and cellular respiration. This is to prepare the cell for cell division. This activation is also triggered by the release of calcium. Enzymes, mRNA, and proteins needed for the activation process are already present in the cytosol of the egg. DNA synthesis will begin and the cell will then divide, signaling the end of fertilization and the development of the resultant embryo. Species vary with respect to the meiotic stage of the oocyte at the actual point of fertilization. For example, human eggs, as seen to the left, are suspended in metaphase 2 until the sperm enters the egg. However, sea urchin eggs complete meiosis when they are released from the female. As stated previously, mammals do not have a fast block or depolarization event after sperm entry. However, they still have an acrosomal reaction and cortical reaction, meaning that the hydrolytic enzymes are still released and the cortical granules will also still be released. In general, sperm must swim through a layer of follicle cells that surround the eggs, as well as the zona pellucida, which is the extracellular matrix of the egg. Once the sperm reaches the egg surface, it can then bind to sperm receptors. The entry of the sperm will trigger a release of enzymes from the cortical granules. This will cause changes in the zona pellucida, which will lead to the hardening of that layer. Sperm must be able to penetrate through the layer of follicle cells and the zona pellucida before it actually reaches the egg. Once fertilization occurs, cleavage will then occur. Also note that the mRNA and proteins that are already present within the cytosol of the egg meet the needs of the early fertilized eggs. Cleavage involves a series of rapid cell divisions or mitosis. This is done to restore the balance between the DNA content and the overall cell size. During cleavage, the cell cycle consists primarily of the S and M phases, which is DNA synthesis and mitosis. This leads to rapid divisions with a successive decrease in cell size as seen in the diagram to the right. This will overall maintain the overall mass of the developing embryo. However, the number of cells that comprise that embryo would rapidly increase. The early cells that result through the cleavage process are termed blastomeres. These will eventually arrange themselves to form a blastula. When a blastula forms, there is an internal cavity that is termed the blastocele. A blastula will form after approximately 5 to 7 rounds of cell division. The cleavage patterns of the developing blastula may be symmetric, as seen in echinoderms or starfish, or asymmetric, as we see in frogs. This diagram here depicts the formation of the blastula stage in an echinoderm embryo. Frogs in general have asymmetric cleavage. This leads to the formation of two different poles, the animal pole, which will eventually house the blastocele, and the vegetal pole, which contains most of the yolk, which will help nourish the developing embryo. As the cells divide, a cleavage furrow will form. However, the unequal yolk distribution will slow cell division in the vegetal hemisphere. This causes the production of fewer and larger cells. The animal hemisphere is usually designated and forms at the point of sperm entry. The diagram to the right depicts the pattern of cell division in the frog embryo. Note the incomplete cell division where the yolk is, and also note the presence of the blastocele, which forms within the animal pole or animal hemisphere. In general, there are two main patterns of cleavage, holoblastic and meroblastic. Holoblastic cleavage occurs when the cleavage furrow can penetrate completely through the egg. This is present in amphibians, echinoderms, mammals, and annelids. Meroblastic cleavage occurs when the cleavage furrow cannot penetrate through the egg due to the high amount of yolk. This is present in birds, reptiles, fishes, as well as insects. In aves, as seen in the chicken embryos to the right, the yolk is considered to be the entire egg. Cell divisions are thus limited to a small white area that is present at the animal pole. The next process in embryogenesis involves morphogenesis. Morphogenesis refers to the cellular and tissue-based processes that lead to the formation of the animal body. Morphogenesis involves two main processes, gastrulation and organogenesis. Gastrulation refers to the formation of the gastrula. This occurs as surface cells begin to move interior. During gastrulation, the embryonic cell layers are established and a primitive digestive tube will form. Organogenesis, on the other hand, involves the formation of organs from these embryonic germ layers that are established during gastrulation. Gastrulation involves the reorganization of the blastula stage into a 2 or 3 layered gastrula. In general, a small group of cells will form a shallow depression that begins to invaginate on the blastula. This will then keep invaginating to eventually form the archenteron. The first opening that occurs at the mouth of the archenteron is termed the blastopore. This will eventually form the mouth or anus of the animal. The archenteron will continue to invaginate and eventually will reach the surface. This will complete the formation of the primitive gut. At this stage, the embryo will have 2 or 3 layers. The embryonic germ layers include ectoderm, endoderm, and mesoderm. Most 2-layered embryos contain ectoderm and endoderm. 3-layered embryos will then have the extragerm layer of mesoderm. Ectoderm typically forms the outer layer of the embryo. This will eventually develop into the epidermis, sweat glands, hair follicles, nervous and sensory systems, pituitary gland, the adrenal medulla, as well as the jaws and teeth. The endoderm will form the inner layer of the embryo. These will eventually lead to the formation of the epithelial linings of the digestive tract and organs, the respiratory tract, excretory tract, reproductive tract, as well as the ducts, and the thymus, thyroid, and parathyroid glands. The mesoderm is a germ layer that forms in between the ectoderm and endoderm. The mesoderm will form skeletal and muscular systems, the circulatory and lymphatic systems, excretory and reproductive systems, except for the germ cells, the dermis of the skin, as well as the adrenal cortex. In general, diploblastic organisms have ectoderm and endoderm present, while triploblastic organisms have all 3 layers, ecto, endo, and mesoderm. Note the difference between the different types of diploblasts, such as the hydra, as well as the triploblast, in this diagram, the planaria, as well as the sea urchin. Note the presence of the 2 or 3 germ layers. In diploblasts, the blastopore becomes the open end of the gastrovascular cavity, thus acting as the mouth as well as the anus. In triploblasts can be termed protostomes or deuterostomes. If you recall, in protostomes, the mouth forms from the blastopore, whereas in deuterostomes, the mouth forms opposite to the blastopore, meaning that the blastopore becomes the anus. We're now going to take a look at the gastrulation process in various types of animals, beginning with the frog. Gastrulation in the frog involves the formation of the blastopore. The blastopore forms as an indentation on the dorsal side of the embryo. This will also lead to the formation of the dorsal lip of the blastopore. This is a region that lies directly above the crease. Cells from the vegetal pole begin to move in and invaginate at the blastopore. This leads to the formation of endoderm as well as mesoderm. Note the presence of the blastocele in the animal pole. Outer cells from the animal pole will then begin to slide toward the blastopore, forming the ectoderm. The archenteron will then form and enlarge as the blastocele begins to shrink. The blastopore surrounds the yolk plug as all 3 germ layers form, ectoderm, endoderm, as well as mesoderm. Note the presence of the archenteron as well, and the remnants of the original blastocele in the blastula. We're now going to take a look at the gastrulation process in chicks. If you recall, embryonic cells will sit on top of the yolk. In early gastrulation, they divide into 2 layers. These are termed the epiblasts, which eventually will form the embryo, and the hypoblasts, which will eventually form the yolk sac. As gastrulation continues, the epiblast cells will move toward and invaginate along the midline. This forms an important structure known as the primitive streak. Some cells will begin to invaginate inward at the streak. This will lead to the formation of endoderm as well as mesoderm. However, some of these cells will remain at the surface, which will eventually form the ectoderm. Note the presence of these embryonic germ layers in the diagram to the right. In humans, gastrulation begins with the blastocyst. The blastocyst is a structure that contains approximately 100 cells that are arranged around a central cavity called the blastocele. Within the cavity lies the inner cell mass. This inner cell mass will eventually form the embryo. Also note that this inner cell mass is the source of embryonic stem cells. Trophoblast cells will line the exterior of the blastocyst. If you recall, fertilization occurs in the oviduct. Blastocyst formation will occur as the fertilized egg moves towards the uterus. Implantation occurs approximately 7 days post-fertilization. Implantation occurs when the trophoblast extends into the uterine lining. Trophoblast cells will release enzymes that help to break down the endometrium. This will allow the blastocyst to invade the mother's tissue. After the implantation process, the inner cell mass of the blastocyst will form a flat disc. This will then divide into the epiblast that forms the embryo and the hypoblast that forms the yolk sac, similar to what we've seen in the gastrulation process in chicks. The epiblast will form the embryo, and it will also contribute to the formation of the extraembryonic layers. At approximately 13 days, gastrulation begins. When this begins, the epiblast cells begin to move through the primitive streak. As seen in the chick embryo, some will stay on the surface to form ectoderm, while some will move inward to form mesoderm and endoderm. At this time, the extraembryonic layers also begin to appear in rudimentary form. These extraembryonic layers originate from the embryonic cells. These include the yolk sac, allantois, amnion, and chorion. Once the gastrulation process complete, the extraembryonic mesoderm and the 4 extraembryonic layers will surround the embryo. As well, the 3 germ layers are fully established. The placenta then begins to form from the trophoblast, epiblast, and endometrial tissue. The yolk sac and allantois will in turn form the umbilical cord. This, combined with the placenta, will allow for the exchange of nutrients, gases, and waste material between the embryo and the mother.